Okay, today we're going to talk about PCR troubleshooting. We will eventually, so throughout this class I've kind of been drawing plasmids and I've kind of been drawing genes. We will, in the next module, discuss actually how to get the gene and put it in the plasmid and how to make the modifications, etc. But much of that happens through PCR. So it's always worth it to talk about common things that go wrong in PCRs and how to optimize PCR, because it's a daily thing that you do pretty frequently as a researcher. Okay. So imagine you set up a PCR in a tube. Okay. And here you run your ladder. So it's an agarose gel. Here you have your sample. Okay. And imagine you run it and you see nothing. This is a common result. Because you do PCR, you see nothing. What could have gone wrong?
Primers didn't bind.
Primers didn't bind. What else? Let's make a list. Let's brainstorm. What could all go wrong?
Bad cDNA quality.
So this is PCR, not RT-PCR. So no cDNA involved.
Could it be too small of a sample?
What do you mean by that?
Like, you put 1 micro instead of 20?
Okay, let's explore that though. So you're right that usually PCRs have kind of like a sample volume.
Yeah.
Usually that's like 20 microliters. But when you say not like too little, like what is not in there not enough? What would be not enough?
Template?
That can go wrong. So like you could have like too little template. But this is, I would say this, well, I mean, it can happen. But because PCR is exponential, like the strength of PCR is that you only need just a tiny, 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 tiny amount. And it can exponentially expand on it. So that can be a problem. We'll keep it on the list. What else? What else can go wrong?
Runs off the gel.
Runs off. That'd be a that's an interesting one to talk about. That can happen. Sometimes you forget about your gel and you just like let it run for too long. But you would know that. How would you know if this would be very easy to detect. How would your gel be problematic if you ran it off the gel? Well, you would actually be able to see something in the gel that would tell you like, okay, I ran it too long.
It would be all the way at the top.
No, if you ran it too long, your ladder would be like very spread out. And so you know that you ran like most of your ladder bands like off, and if your PCR band is down here, then okay, then you're like, yeah, okay, I ran it off. So that'd be a pretty easy thing to detect. What else can go wrong?
You used the wrong thermocycle?
Like the program?
Yeah,
Like
you didn't have the correct intervals.
wait, so, I'm sorry, I didn't get that. You didn't have the correct what?
Intervals.
Intervals.
And like temperatures and stuff.
Oh, okay. So I'd call that like your program is wrong. Yeah. Yeah, that can happen a lot. Okay. So we understand like these are some of the things that can go wrong. There's more. We understand that there's a lot of stuff that can go wrong. If you run a PCR and you don't see anything, what should be your troubleshooting pipeline? Like what should you do right away?
A second PCR.
Yeah, actually that is usually like what I always say. If a student does something and it doesn't work, I always will say, do it again, because usually the first time you do something, like you fail in science. So that is a good one, like just do it again. Although before you just do it again, like there are some things you should check. So we should check positive and negative controls. So every time you run a PCR or any experiment at all in science, there should always be a positive and a negative control. So the fact that I drew this with a ladder and just a sample, that's a bad experimental design. You should always have a positive control and a negative control, okay, before you test your like sample that you're running. Okay, so why do you have a positive and a negative control? Like what will they tell you?
A negative tells you if there's contamination.
Yes, okay, so oftentimes, okay, good. So oftentimes a negative control is either water or depending on what like you're checking for, it could be DNA that will not produce a band. Okay, so if you saw a pattern that looked like this, where you're getting a band in your negative control, then you know that all this data is flawed because something is probably contaminated. So negative control is going to tell you about contamination. This is very common. This is a common problem in PCR where you might be like working in a lab, you'll be doing PCRs, they'll be working for you, and then all of a sudden everything that you do is starting to become positive. Like you're getting PCR bands in every PCR that you ever do. What does that suggest? What's contaminated? Probably.
Your reagents?
Yes. Okay. So let's review. What are the reagents that go into the PCR? And let's think through these. What goes into PCR? Primers. Template. What else? Buffer.
DNTs,
DNTPs.
water, or special water.
Yes. Okay, so the most likely things to become contaminated are, well, many of these things can become contaminated. Water is like the most common thing. So when you are running PCRs, you'll typically run PCRs when you mix up the tube. You have like a special water at your station. And typically like that water comes in kind of, like you'll buy your molecular biology grade water, and it'll be in like a container like this. So one of the things you never ever want to do when you're running PCRs or doing molecular biology in general is you never want to take this big jug and then open the cap and then take your pipette and like take water out of this thing and then close the cap and then put it in your PCR. Why do you not want to do that?
Your pipette could be contaminated.
Yes, and usually it is, like pipettes are dirty. So you should never ever take a giant container of reagent, open the cap, stick your pipette in, and take it out. Usually what you do is you make what are called aliquots. So usually you'll set up like some tubes that you can have at your workstation. You'll open this up, you'll pour it into some aliquots, carefully pour it so you're not like getting anything in it, close it up, and then put this up on the shelf, and then these you can close and have on your bench, okay? And when you're running PCRs, you'll typically be drawing from your what are called aliquots, okay? And after you've used an aliquot a few times, the reason it's an aliquot is because you should just throw it away. So after you've used this reagent, like enough times to the point where you worry it might be contaminated, you should just throw that away and go to the next one. Use that for a little bit, throw that away and go to the next one, etc., etc. Okay? So this is kind of an important scientific thing, aliquoting. You also do aliquots with many of the reagents. So when you buy DNTPs, and this is to prevent contamination, okay? So when you buy DNTPs, you'll get like a cryotube, a vial of DNTPs, okay? And then usually you will set up aliquots, and as soon as you get this, you'll open it up, you'll take out 50 microliters, put 50 microliters in here. Take out 50 microliters, put 50 microliters in here, etc., etc. Close these up, put them in the freezer. And this is a tool to help prevent contamination. You never want to be drawing from the same source over and over and over amongst like a whole lab of different people. Okay, so that can help you prevent contamination. Primers often get contaminated. So when you get primers, when you order primers, after you've designed them, you order them, they come in a little tube where it's dried powder. Okay, and then you have to add water to that. So a common thing where people will introduce contamination is when they add water to their primers, they're adding from a contaminated source, and then any PCR that you run from that is obviously contaminated. Okay. So, etc. Like lots of things can get contaminated. Keep that in mind. If you ever see this result, that's indicating there's contamination. Okay. What if you see this result? What would you think? What went wrong here, and what could have possibly been the mistake? So what's the problem with this gel?
There's no band on positive.
Yes. Okay, so you're missing a band, which should be here, but it's not. So it could be a lot of things that make your positive control go bad. So this could initiate a giant list of trying to figure out like what's wrong. But oftentimes, before you do any PCRs, there's a genomic extraction where you're extracting DNA. Okay, so if you ever have a result like this, probably your process of DNA extraction is bad. If it's not that, then maybe it's downstream PCR things. But if your positive control is missing, then perhaps your process of extraction is bad. Okay. So let's further elaborate on this list of things you should do. Let's see. Okay, so I would first check for positive and minus controls in your experiment. Then I would check the primers. Check the primer sequence. That would cover this. So if a primer doesn't bind, usually the biggest mistake is just when you design these primers, you're looking at some sequence of DNA on the computer, it's a text file, and you're picking out some primer, and usually you just make a mistake. Maybe you type out something and there's some mistakes. Or maybe you forgot to flip a primer in the right orientation. Okay, that's a very, very common mistake. So check your primer sequence on the computer. That's a very quick thing you can do. Okay. Then I would, to do what Alex said, I would just do it again. Do it again, see if you see the same result. Usually, sometimes if you just do it again, things miraculously like fix. Has that been your experience?
Yeah, definitely.
Yeah. Like molecular biology is very kind of like religious like that. You try something and you fail, and sometimes if you just keep trying, sometimes it just works. Okay. Now let's assume we checked our primers, and they're okay. And we're able to do a positive and a negative control successfully. Okay, so it's not our DNA extraction, it's not our ability to set up a PCR, everything's working, but we're not getting a band. The next thing that you might try is what's called a gradient PCR. Okay, so what is it? Gradient PCR. Okay, so gradient PCR, you have a thermocycler. Thermocycler kind of looks like this. Okay, and this is the heat block where you put your tubes. Okay, and this is what cycles the temperature. Okay, and then there's a lid. So what a gradient PCR is it's a fast way to test a whole bunch of different annealing temperatures. Okay, so gradient PCR tests annealing temperatures. Oh, you spelled that wrong. And this is probably one of the most common fixes for a broken PCR. Okay, so my first question is, how do you know what the annealing temperature should be set at in the first place? Like when you're setting up your program, okay, so we have initial denature, which is usually 2 minutes, 98 degrees Celsius. And then we usually have the loop, which is 98 degrees Celsius for 10 seconds, denature. and then you have to pick some temperature X, which you might not necessarily know, for about 30 seconds annealing. And then you do 72 degrees Celsius for a set amount of time based on the length of your amplicon extension. And then remember this loops 35 times. How do you find that value?
The number of base pairs in your primers.
Yes, so it's the A's, T's, G's, C's in your primers. So there is a computer algorithm, which I'll show you later in the next module, where you just take your sequence, you put it in the computer program, it looks at the A's, T's, G's, C's, it tells you what the annealing temperature is. But because there's two primers, so you usually have a forward and a reverse, we should talk about this. So let's say this one was 56 degrees Celsius, and this one was 60 degrees Celsius. Which temperature should you choose for annealing and why? Any guess?
60 degrees,
Why?
because at least it hits 50 degrees Celsius and 60.
So I think about it opposite. So my rule is always pick the lower temperature. And the reason is because if you anneal at 60 degrees Celsius, what's gonna happen with the lower primer? Is it gonna be a looser bind or a tighter bind? So heating things up makes them pop apart, right? And so 56 is the exact temperature at which this thing will bind pretty tightly to the DNA. So if you go above that, what will happen?
It'll separate.
Yeah, it'll start to loosen up, okay? So I always choose the lower of the two because you know that this one can bind at that temperature, whereas this one cannot bind at that temperature, in theory, okay? So you always choose the lower of the two. So as a first guess, I always put the lower of the two, okay? And usually that works, like 70% of the time that works. 30% of the time you get nothing and nothing works, okay? So to optimize this, you can do a gradient PCR, which quickly tests a whole bunch of different temperatures for annealing. And sometimes weird temperature combinations work for whatever reason. And it's not always like readily explainable rationally in a logical sense. Okay, so what a gradient PCR does is in most of the advanced thermocyclers, you can tell it to set a gradient where you tell it, okay, on these ones, at this bottom row, do the annealing at 50 degrees Celsius, and on this top row, do the annealing at 60 degrees Celsius, and on each of these rows in between, do a gradient where you're increasing the temperature from 50 to 60. Does that make sense? Okay. And you could, then what you would do is you set up I think there's usually about 9 rows, something like that. So you would set up 9 tubes of the same PCR reaction. Okay. Same like ingredient mixtures, etc. Then you load those into the thermocycler heating plate, program a gradient, and then you just run it, okay? And then you would run a gel from the results. So once done thermocycling, you take it out, you run an agarose gel. Let's say 1, 2, 3, 4, 5, 6, 7, 8, 9. So this one is 50, this one is 60. Maybe you'd go higher, maybe you'd spread, or maybe you'd go lower. You can spread the ranges pretty far. So that means this one in the middle is 55, etc. Okay, and you'd run a gel, and let's say you saw a pattern that looks like this. What would you conclude? What's the best annealing temperature?
55.
Yes, okay. So this is a very, very fast way to quickly screen through a whole bunch of different annealing temperatures. And in my experience, this fixes most PCRs like very quickly. you'll find a good annealing temperature via this mechanism. Oftentimes it's something really weird, like you might see a pattern that looks like this, which for some reason suggests that your primer pair needs to be cold at annealing for whatever reason. Or you might see something different at the hot temperature, okay? But this is a fast way to cycle through, cycle through gradient temperatures. Okay, so that's called a gradient PCR. Okay, let's do some other things that you commonly see. If your PCR still is not working after gradient PCR, then I try what's called a hot start PCR. Okay, and now I'll talk about primer dimers. So a hot start PCR is a special program that fixes a few common problems with PCRs. So if you have, let's say, a ladder, okay, and let's say you have your positive, negative, and your sample, and let's say you see something like this. This is a very common result of an agarose gel where you do a PCR. Let's say this is the 100 base pair ladder. You see this band, very, very tiny, like in size, on the bottom of the gel. Does anybody know what that is? So sometimes when you do your PCR reaction, sometimes your primers have little sites where on accident they are able to bind to each other, okay? What kind of a problem is this gonna cause when you try to run a PCR? What's gonna happen? In the tube, like envision the molecular biology. What will happen if your primers are doing this?
Well, the primers will just bind to each other, not to the DNA template.
What will get written? Like when the polymerase comes on and it does the extension step, what will get written then?
The primers.
Yeah, the primers. Okay. So when the polymerase, if this is happening, instead of binding to your template, that's DNA, instead of binding your template, if your primers are doing this, when the polymerase starts the extension, it's gonna write this and this. And this is gonna produce a little tiny band, that's a tiny size, okay? That runs at your bottom of your gel, and this is called a primer dimer. That's a super common problem, happens all the time, but it has a really easy fix, okay? So the fix is the hot start PCR. So in the hot start PCR, let's do our program again. We have initial denature, 2 minutes, 98 degrees Celsius, and then we have our loop. We have our denature, anneal, extension for various values and various times. Okay. So when you do a hot start PCR, you add a step right here, right before you enter your loop. And what you do here is you set it to something like 85 degrees Celsius, which is relatively hot. It keeps things pretty much completely denatured. And then you add your polymerase. So in a hot start PCR, when you mix up your tube of your reagents, 1, 2, 3, 4, 5, 6, one of those reagents is a polymerase. The Taq polymerase or Fusion or Q5. In a hot start PCR, you don't add the polymerase when you're mixing up the tube, okay? You leave the polymerase out. And you close your tube after you put in the other ingredients, you put in the thermocycler, and you run this program. It does an initial denature at 98, and then when it hits this step, 85, you can press the pause button on the thermocycler. You can open it up, you pull out your tube, you open it up, and then finally, at that point, only at that point, you add your polymerase. So in a hot start PCR, your polymerase is not going into the tube until when the program is right about to start the loop. How is that going to change the effect of primer dimers? How would that change a primer dimer result? Yeah, so if you mix up the tube when it's cold, like or at room temperature, and you add the polymerase, then the polymerase can just write those primer dimers right away. If you don't add the polymerase until the reaction is hot, which is why it's called a hot start, what's gonna happen to the primer dimers when it's hot?
They'll separate.
They will separate, okay? So they'll be separate. And then you add your polymerase, so it's not writing that. And then you enter the loop, and it will usually succeed, okay? So a hot start fixes one problem, which is the primer dimers, usually. Okay, another problem that the hot start will fix is a hairpin. What's a hairpin? So if you have a primer, and sometimes in PCRs, primers get real long if you're doing weird stuff, okay? And a hairpin looks like this. So a hairpin is where one sequence in your primer can bend around and anneal to another sequence in your primer to form a little region of double-stranded DNA right here. Okay, that's a problem very similar to the primer dimers, because if you add polymerase right to this, the polymerase will see this as a primer and it will write and it will destroy your primers. Okay, so this is another common problem, these hairpins, that the hot start also fixes, because if you don't add the polymerase until it's hot, when it's hot, the hairpin will be released. Okay, so this hot start is a really clever trick that's really easy to implement that usually fixes a lot of problems with PCRs. Okay. Other things that can go wrong, or other things that I would do. So another thing that I commonly do is test template dilutions. So somebody brought up, it's possible that you put in too little template. That is a possibility. Although in my experience, what usually causes a problem is way too much template, okay? What would be the problem if there was too much template? So you have your DNA that your primers bind to. What's the problem if you have way too much of this? Can anybody visualize what the problem is? Why it would not generate a successful PCR?
The primers have a hard time finding the binding spot.
Yes, okay, that's one. So if you, for example, if you have extracted DNA from an entire organism, you have that entire organism's entire genome, and many primers have multiple sites that they can kind of like bind to, okay? So they might have a real difficult time where the forward primer has to find and the reverse primer has to find, okay, to get the actual amplicon at the right size. So having too much DNA, that can make it harder for the primers to find their spot, okay? So what I often do is I test template dilutions. So if you have your template, your genomic extraction in a tube, okay, what I will do is I will make a 10x, a 100x, and a 1000x dilution. So dilutions are very common in molecular biology. Hopefully this is not hard for you to understand. How do you make a dilution? What would you do? How do you make a 10x dilution? Let's say you have 100 microliters.
You take 10 microliters,
And do what with it?
put it into the 10x,
And then what would you do?
put 90 of DI water.
Not DI water.
Or-
Use like the molecular grade water. But yes. Okay, so that's how you would make a dilution. So you do 10 plus 90 microliters of super high grade water. And then once that's mixed up, you can iteratively do the same thing down the row. Okay. And then what I would do is I would run 3 PCRs with these different templates. And oftentimes you might get a reaction where this one is producing a band, but this one is not. Okay. That's a common fixed PCR. Okay, template dilutions. If you get to that point, if you get to this point, you've done all these things and your PCR is still not working, I would redesign the primers. So oftentimes when you're running a PCR, here's your GeneX. Imagine you were gonna PCR GeneX. Maybe you designed your primers here and here. Usually you have some flexibility in designing where these primers anneal at. So sometimes it's as simple as you got unlucky, and if you just scratch these primers and you just pick different sites where they're annealing, all of a sudden now it'll work, okay? That's a very common fix. Just redesign the primers, try it again. And primers are very cheap. So PCR primers for a forward and a reverse cost about $10. So it's very cheap to just quickly get a few different sets and see what's working, what's not working. Okay, are there any questions about, this is kind of like troubleshooting PCR, what you might do? Any questions about that? And then if not, we'll do some more examples. How much time do we have left?
19 minutes?
19 minutes. Okay, let's do some. Let's do some examples of like things you might get asked or about, or you should be able to handle. Okay, so let's do example problem 6. Detect the presence of protein X. Okay, so here's the experimental conundrum. Say we clone gene X into a PBAD plasmid, which is an arabinose promoter. Okay, if we did this, the goal of doing this, what's essentially the goal of putting gene X in there, in this plasmid in particular? This implies that it's being used to express the protein. So the goal is to essentially like add arabinose and produce protein X in mass quantities. So if this is the goal, what you would actually need to know if this was working is, can you detect the presence of protein X? What experiment would you do to see if this is working? So maybe if you're confused, first ask, what is the type of molecule that we need to detect? The protein. So you're working with then a subset of methods that apply to proteins. What can we do with proteins?
It's like tag them with a fluorescence.
But I don't want to change the construct. I want wild type.
PCR or not PCR?
PCR is analyzing the nucleic acid, not the protein.
SDS page?
Yes. Okay, so we would run a gel, run SDS page. Let's say here's our ladder. Let's say the size of protein X is 25 kilodaltons. It's KDA. That's a unit of size for proteins. So let's say here's our 25 KDA marker. What's going to be we need a positive and a negative control. And then we have our test sample. What's going to be our positive control? Or what could we use as a positive control? Think outside the box. Any ideas? This is often like a common scenario where you're asked to do an experiment as a graduate student, and it's your job to figure out what is a positive control.
Do I have protein X?
In this case, no, because you're trying to, that's a good question. In this case, no, because you're trying to produce it for the first time. You're saying you could just take protein X and run it in the sample, if you had it. Some cases you might have that. You might actually be able to buy like protein X. But then if you can buy it, it's like, well, why would you try to produce it? So what could be a positive control? You look like someone was just about to come on. Sometimes you have to build positive controls. Could you build a positive control? No guesses, even from you guys?
Could you just a gene X in a plasmid already?
Well, if you already had gene X in another plasmid, then it's like, why would you make a ditto, a different, like subtly different version of it?
Would you just find a protein of similar size and run it?
That's pretty close. Okay, so that's pretty close to like on what you might do. So oftentimes when you buy sets of backbones, so when you acquire plasmids, oftentimes they'll actually send you like positive controls as part of the packet. So sometimes you'll get sent a backbone plasmid that has a protein called LAC-Z. LAC-Z is an important protein from the lac operon, beta-galactosidase. It's often a positive control in many different experiments in molecular biology. So if you have a plasmid that looks like this, okay, you can use this as a positive control. What's the positive control verifying? So in this case, you would add arabinose and it's producing LAC-Z. I think LAC-Z's size is something like 120 kilodaltons. So you would see something like this. So what's the positive control confirming in this case?
Your transformation worked?
Yeah, it's confirming your transformation, it's confirming your arabinose induction, it's confirming your ability to do an SCS page and your protein extraction. Okay. So, and I should actually be more careful. Probably what you would see is you would see something that looked like this. You'd see a ton of protein band. If you didn't do any advanced purifications and you ran essentially like global E. coli protein, you would see a ton of protein bands, okay, that represent the global constituents of protein in the cell. And then you would see probably a really, really abundant band where it should be at about 120 kilodaltons in positive control, okay? So then what is your negative control? What should be your negative control? Or what could be a negative control?
Just the backbone?
Yes, that'd be a good one. Okay, so in this case, you have just a sealed backbone, nothing cloned into the multi-cloning site. It's just empty. You add arabinose, nothing would get turned on. That'd be a good negative control backbone. And if that was the case, you would see this. The pattern of global proteins in E. coli would look very similar to your positive control, except it's missing that LAC-Z, okay? There's actually another really good negative control. What would be another negative control using this construct?
No arabinose?
Yes, that's the most useful negative control, is you run the experiment, except you don't add arabinose to induce. Then you're gonna get similar. It's gonna look like this. The pattern will be the same, okay? But you have not induced that protein. And now run your sample, okay? Your sample, in this case, plus arabinose. So sample plus arabinose. In this case, you should see all these bands plus something right at 25 kilodaltons. You might see something that looked like that. This would be a positive confirmation that your protein expression is working. Does that make sense? Okay, it's a good experiment. We'll talk more about that in protein expression. How much time do we have left?
10 minutes?
10 minutes, okay. Let's quickly go over, since it's come up a few times, let's go over RT-PCR. Okay, so RT-PCR is reverse transcriptase PCR. So what is RT-PCR used to study? RNA. Usually expression. So oftentimes, like, the context under which you're doing RT-PCR is some kind of an experiment where you want to know expression. So maybe you have a question like, let's call this like example 7. Maybe you have a question like, design an experiment to determine which life stage of fruit fly, Drosophila, expresses, what's a cool gene? Vitellogenin. Okay, so here's an important protein for having babies in Drosophila. It's an egg yolk protein, so it's important for them producing eggs. So I'm saying to do an experiment, design an experiment, to determine which life stage of Drosophila melanogaster expresses vitellogenin. So what you'd have to do this is you'd have to take the different life stages. So there's an egg, there's different larval instars, okay? These are like little worms. I don't precisely remember how many instars there are. Let's say there's 1, 2, 3, 4. And then there's a pupal stage, and then there's an adult stage. Okay, and you want to know which stage expresses vitellogenin. This is a very common scientific question. When is this gene being expressed? When it's expressed, gives you some information about what it's doing to the organism. Okay, so in order to run a PCR to test expression, what do you need to purify? RNA. Okay, so you'd run some kind of a, like I'm listing out like the steps. You'd run some kind of a RNA purification at different life stages. So you'd have one tube where you're purifying from eggs, different larval instars, pupae, adults, etc. You do your RNA purifications. Okay. I'm trying to think of like how much detail I should give. Then the next step is the reverse transcription. So the reason that you do this reverse transcription, actually let's review. What's reverse transcriptase? What does RT do? What is it and what does it do? I talked about this under the context of the central dogma, where I said this is the one enzyme that breaks the central dogma. So what's it do and what is the name of, what type of enzyme is it? It is a polymerase. It's a RNA dependent DNA polymerase. Okay, so what this unique enzyme does is if you have a transcript, here in this case for vitellogenin, okay, what RNA polymerase does is if you design a primer that binds to the 3' end of a transcript, so messenger RNA, what RNA polymerase can do is it finds this, okay, and it will write a matching sequence of DNA. And this DNA has a very specific name. It's called complementary or cDNA. So cDNA is the complementary strand product. That's what's produced that binds to the messenger RNA that's made by reverse transcriptase. Okay. So why do you do this? The reason that you do reverse transcriptase is because RNA is not very stable. It's actually very difficult to study because there's a whole bunch of enzymes that degrade it, and it just kind of collapses pretty quickly. It's not very stable. So if you can build this, you've essentially like stabilized the transcript. So if you have cDNA, it's now stabilized in a more stable construct where you can do various things to it. Sometimes people will sequence from this, which is called RNA-seq. Sometimes people will immediately put this into a library, into a plasmid, clone it into a plasmid. And otherwise, people will do RT-PCR, which is, so right after the reverse transcription, you can take this becomes your template, the cDNA becomes your template, and then you do a PCR, which in theory you've designed primers that bind inside of this sequence. And then you do a PCR and you would produce an amplicon. So you'd run a gel. Okay, so here, I'm gonna erase so I have enough room. Everybody got that, hopefully. So in an RT-PCR reaction, in this RT-PCR reaction specifically, you run a gel. You're gonna have a ladder. It's an agarose gel because in the end you did a PCR which produces DNA. So you're actually no longer studying RNA. You've produced DNA that came from a source of RNA, which you converted to cDNA, okay? And you run your samples. So let's say you ran your different life stages, 1, 2, 3, 4, 5, 6. Let's say this one is adult, okay? And depending on the size of your amplicon, where your primers bind, let's say it's 200 base pairs. Here's 1, 2. Let's say you saw a result that looked like this. What's your conclusion?
Adults best.
Adults express vitellogen. Okay? So RT-PCR can give you a visual readout of expression of a gene, some gene X. Any questions about that? What time is it? How much time do I have now? We're done.
4 minutes.
4 minutes, we still have time. Okay. So then let's define RNA-seq. Okay, RNA-seq. Well, I mean, there's different ways to do RNA-seq, but oftentimes what's happening is RNA-seq, it utilizes a reverse transcription step. So in RNA-seq, which is also used to study expression of genes, you would do the RNA purification, you would do the reverse transcription, and then in this case, oftentimes different things happen. Sometimes you would make a library. So perhaps if you do RNA-seq, I'm thinking of how best to explain this. RNA-seq would answer the question of design an experiment to determine when all genes are expressed. Looking at every single gene in the genome, the entire set of genes. That's what RNA-seq is capable of. So you would still do an RNA purification of global RNA. You do reverse transcription reactions, but in this case, the difference between RT-PCR and RNA-seq is in RT-PCR, you designed a specific primer that bound to the 3' end of your vitellogenin gene, which you were targeting. You were looking at a specific gene. In this case, you're not looking at a specific gene, you want to look at all genes. So what kind of primers do you need to be able to make cDNA from all possible genes?
Degenerate?
I wouldn't use that word. I think what you're saying is like, you're probably saying like, what would be the word for that? You're essentially saying that like they sloppily bind everything. So there are primers that, those are called random hexamers. So random hexamers are just a series of 6 A's, T's, G's, C's randomized, and they can bind all different transcripts because in theory, all different transcripts have different sequences of all random sequences. So random hexamers is just an assortment of random primers that sort of distribute across all the RNA and bind to different RNA transcripts at different spaces. That's one source of primers that can give you the reverse transcription to sequence all genes. What's the other one that you can use if you are in a eukaryote? How is eukaryotic RNA unique from prokaryotes? What's right over here? Eukaryotic messenger RNA. Yeah, so in eukaryotes, they have a poly A tail. All transcripts have a poly A tail. How do you design a primer to bind to all messenger RNA then?
Poly T-tail.
Yeah, so that's another primer. It's a poly T-tail. It just binds to all messenger RNAs that have a poly A tail. And then your reverse transcriptase can bind here and produce a product, cDNA product of all the messenger RNAs. Okay, and then once you have that, oftentimes you'll make a library. So oftentimes you'll digest with a restriction enzyme, in theory, and then you can clone these little chunks into plasmids, and then you can sequence all the plasmids with like Illumina. this would be a pipeline for RNA-seq, one example. So you should know how RNA-seq is different from RT-PCR. RT-PCR is using a specific primer, binding a specific messenger RNA. RNA-seq is making cDNA of all possible transcripts and then doing high throughput sequencing to give you a list of all the genes that are expressed and the expression levels. Any questions about that? Good, okay, that's it. So you guys can go to the lab now and you'll set up your spots, your spot assays, and we're gonna see who got stuff that will grow.